Okay, so let's start this. No, it would be okay if I stay here because I'm sort of too much furniture over there. Okay, so this will be the course about comparative genomics and what people do in this area, why it's interesting, and I mean, why it's useful, at least in some sense. And probably you've heard something about some of the things I will be talking about in your applied informatics courses, but I'll still repeat it for completeness and because I do believe that students normally forget everything that have been told in about a couple of days after having the final exam, so it wouldn't be boring at least. Okay, now I do know that students both at Caltech and High School of Economics are extremely concerned about formulas for calculating their final grades. I just read the responses of students' questionnaires, both in Caltech for all subjects, because I'm the head of the master's program was here and at Caltech, so I read lots of these responses, and the most frequent one was that it wasn't clear how it will be graded at the end. So I'll start with the grading scheme. The grading scheme would be as follows. You'll have several major homeworks, something that would require some programming or some research or whatever. And I'll grade it. I'll grade these homeworks. I'll try to grade them as soon as possible. I mean, immediately after more or less immediately after the deadline. And then you'll have some feedback on that. Secondly, there will be some questions arising during the lectures. I don't know beforehand what they will be. That depends, I mean, on what we are discussing. Those will be simple ones, requiring 10 to 15 minutes of deliberation during your whatever spare time. I would expect getting responses to these questions by midnight from Saturday to Sunday of the week, and you'll get some small points for this as well. If you need an exact numerical formula, I can invent one. But one thing you should actually know is that any professor can grade any work with any grade, and it would be absolutely fair, and you'll get whatever grade the professor wants anyhow. So all this fuss about formals actually is done. Anyhow, so this will be the bulk of your final grades. I will announce your preliminary grades like the last lecture or slightly before that. I mean, I will send you letters. I won't read it aloud. I'll send you personal letters with your preliminary grade. If you have done everything well, then you'll get the highest grade, and that's it. If you miss something or they're not perfect, you'll get some grade, and then during the exam, you may improve it. So if you are satisfied with your grade, that's it. If not, you come to the exam with talk, and you have this opportunity to improve your grade, your total grade collecting during the term. One more thing that I forgot to tell is that we'll have a regular journal club. I mean, I do know that you have had lots of journal clubs already, but the style of a journal club is different and the substance is different. It's actually important because you have an opportunity to read really new papers that have been published this year. last year, more exactly. And then we'll discuss that. And the journal club would be graded in the same way as your major home projects. It would cost, well, approximately the same. Yeah, the exam. The exam is just a discussion. I don't have, I mean, if you're trying to upgrade from, I don't know, D to C, you basically select whatever topic you like and convince me that you know at least something. If you want to upgrade from C to B, you list several topics and I select one that I like. But not necessarily. If you want to upgrade from B to A, then I just select what to discuss with you. So it's more or less like that. It's not, I mean, again, I do know that students prefer to have a list of questions that are just to learn them by heart. But again, first of all, I'm too lazy to prepare this list. And secondly, it's so extremely boring that, I mean, I start hating it immediately, and then you have no chance to improve anything because I hate you from the very start. So we won't do that. Instead, yeah, a typical question during the exam, well, two typical questions during the exam, again, to upgrade from B to C is one of the possibilities is that I ask you to recall the paper that you have prepared for the journal club, to recall at least something from that paper. I mean, why have you selected it from the very beginning or something like that? If it's from C to B or from B to A, I might ask you to recall the most interesting paper you've heard about at the journal club from somebody else. So this is, to some extent, it's my tool to convince you to attend the journal club and to listen what's going on there, at least from time to time. Today we'll have no journal club, of course, because you haven't prepared anything. Now, for the journal club, what are possible topics? Basically, it could be, well, actually, I would like to leave it. I would like to leave the selection of papers to you. So what you do, you like read abstracts in whatever area you're interested in, but somehow connected to comparative genomics, or it might be systems biology, something to do with like omics analysis, if it's really interesting as models. It should be published in, well, if it's earlier than 2017, so if it's 2016 or earlier, or earlier, then I would specifically ask you why you think this paper is interesting. So basically, we select a paper published in 2010. If it's really interesting, if nothing more interesting or more substantial have been published on the topic since, but then it's actually suspicious because if nobody has written anything about the topic in 9 years, then probably the topic itself is sort of boring. So be careful with that. But then, I mean, you need some specific explanation why you think that this paper is still interesting and actual. So by default, it's 2017, 18, and this year. Again, I consider comparative genomics as a rather broad field, so it shouldn't be limited only to the topics we discussed during the lectures. And it actually might be good if you think of something like interesting slightly beyond the boundaries that I had during the lectures. For instance, and then if you find a paper on using transcriptomics for comparative analysis of gene expression during embryo development in mouse and I don't know, frog, that's fine. I wouldn't touch that topic during the lectures, but it's clearly comparative transcriptomics in a broad sense. It's likely to be interesting, so it's okay if you select something like that. What you can do, you can send me your ideas about what you would like to prepare, and then you'll get my feedback whether I think that this paper is okay, or I think it's not interesting, or maybe I think it's not related to what we are doing. So you may use this opportunity actually to consult with me on what you're proposing to discuss. My email, probably you know that, but just in case, it is. Sorry. Okay. What else on organizational methods? One moment. Yeah. We have actually lots of people. We have about 20 people from 19 more exactly from high school of economics, and I think something like 6 to 7 probably. No, more from Colcat. Okay, I see 6, 7, 10 from Colcat. So we have 30 students, which means that we'll have to have something like 4 presentations, maybe even 5 presentations that day, which is a lot. So okay, let's double it. Okay, let's do it this way. Let's do it in pairs. So every 2 students, I think I can do 4 pairs, and each pair prepares a paper. Basically, it's up to you how you divide the labor. But in this case, I do expect that you read it really carefully and try to find like bugs or whatever inconsistencies or whatever you don't like. So this your like you have somebody to. It might be a good idea, for instance, to form pairs in such a way that one of you thinks that the paper is good and presents it in the best possible way, and the other one thinks the paper is bad and tries to find faults in that. And then we have like competitions and then trials, and that would be fun. If not, as I said, it's up to you how you divide the labor. So you will have 15 presentations, and which means that if we have 3 presentations per day, it's half an hour for a presentation, which is sufficient time to discuss in real depth. So you'll have like 10 minutes to present the most interesting things that are there, 10 minutes for me interrupting you during the presentation, and then 10 minutes for the final discussion, which is fast. So you prepare approximately 10 slides for your presentation, give or take, which means that you present only the most interesting findings. So you tell the story. The idea is that you don't go like from figure to figure, reproducing everything that has been written there. It rather means that you tell why the paper is interesting, what's important, what findings you think are important, what could be improved or what has been done badly, what you think might be further development in the area. So it's not just like you're retelling the paper, you're preparing your own presentation based on what you have read. If you read something beyond this paper, to put it in some context, it's clearly a bonus. And then we need to organize that. So I suggest that somebody creates a Google Doc in Excel form or something, Google spreadsheet or whatever. And then you just you write your names there, the names of both members of the pair, write there the title of your paper and at what day you want to present it, keeping in mind that we have 3 presentations per day. Could you organize yourself in that? Do you have a common email for everybody in this course? Have a common mailing list. Okay, then again, another request is that somebody creates a Google mailing list, and I'll ask my TAs. Well, we'll do it the other way around. I'll just ask my TAs to make this mailing list, and that's it. Okay. Cool. And actually, I'll ask them to do the spreadsheet, and we'll just send you. I just forgot for a moment that I can case. Sorry. I love them. Okay, good, don't bother. And we actually do have your email somewhere in Canvas, so you'll get this information. Yeah. Okay, let's go. Okay, that's it. Any questions about like logistics? Okay, so I actually have a request about the logistics, which is that when you become tired or bored or deprived of oxygen or whatever, you tell me and we make a break. So I wouldn't follow the clock. You tell me when you think it's time for a break, whomever, and then I'll go to the closest logical stop. Stop there. We have like 5 or 10 minutes. Then we return and go on. So again, the breaks is up to you. Okay, so let's go. Well, the basic stuff of comparative genomics is, of course, BLAST. Actually, I think that you have not seen this screen because it's very obsolete. It's how BLAST looked like, I don't know, 15 years ago or something like that. I keep it both for its sentimental value and also because actually nothing has changed much. So you have all the basic BLAST queries here, and of course what BLAST does, it finds your sequences, data sequences or protein sequences related to your query. Now, what's wrong with what I just told you? Specifically, I intentionally have misled you in my last sentences. In what way? Come on. Well, the idea is that similarity, sequence similarity, does not necessarily imply homology, does not necessarily imply common origin or whatever, because proteins may be, or nucleotide sequences might be similar for several different reasons. One of them is, of course, that they have that they are homologous, that they do have common origin. But it also might be that proteins, for instance, have skewed nucleotide composition, like this one. It's a real protein. It's expressed. You might immediately spot that it doesn't look very much like protein. It looks rather like cat was sitting on the keyboard or something, or walking around the keyboard or something like that. And there are lots of proteins like that, or there are segments like that in many different proteins. So, for instance, here you have lots of glutamate, lots of Q, glutamate probably. Q, which amino acid it is? Oh, I honestly don't remember, but you should. Okay, well, lots of glutamate. Glutamine. Okay, so there are lots of glutamines here, thank you. And this protein would be similar on the sequence level to any other protein that also contains a lot of glutamines, as we do know that there are lots of proteins like that. It's absolutely not necessary that they have common origin. So similarity may arise from many different reasons. One of the major reasons is the skewed amino acid, locally skewed amino acid composition. All glutamine-rich proteins would be similar, or proteins containing glutamine repeats would be similar to each other on the sequence level, and so on. So what you get similar sequences, but not necessarily related sequences. That's one thing that you have to keep in mind. And there used to be a very nice story that I used for many years to illustrate this point, but it's not working anymore, probably because something has changed in BLAST settings. So I'll just tell you the story as a historical anecdote. So historically, like 5 years ago, if you did BLAST X on any GC-rich prokaryote, you would get lots of collagens with quite significant E value. So BLAST X formally translates a nucleotide sequence into an amino acid sequence in 6 reading frames, 3 on the direct strand and 3 on the complementary strand. And if this initial nucleotide sequence is highly GC-rich, like 70% GC-rich, you would have, just due to the structure of the genetic code table, you would get a protein, or rather amino acid sequence, highly enriched in glycines and prolines. And we do know that collagen is proline, glycine, sorry. And again, proline, glycine, actually. And so what you would get a glycine, proline, rich amino acid sequence that would be highly, significantly similar to lots of collagens. Of course, there is no biological relatedness in that. And now I've asked my students to do this trick several years ago, and it turned out it's not working anymore. So probably something has changed in BLAST settings. It's likely that they have improved their procedure for filtering these low complexity segments. So what BLAST does, actually, when it looks for similarity, it filters out segments with highly skewed amino acid composition or low complexity segments that they call. But then, in some cases, there is basically nothing left. So, okay, similarity does not, homology is common origin by definition. Sequence similarity does not necessarily imply homology. But even if two sequences are similar, and even if two sequences are homologous, it does not necessarily imply that their function is the same. Because this partial homology, it might easily happen that only some domains of proteins are homologous, and the remaining domains are different, of different origin, and we cannot, in this case, you may not transfer what you know about the function of one protein to the other protein. Now, what is a protein domain is actually a nice concept, because it's very badly defined. Everybody speaks about protein domains, but in many cases, people actually don't agree on what these domains are. There are at least 3 different approaches to the definition of a protein domain. So one approach is biological, like functional. Protein domain is like part of the protein that has its own independent biological function. So you may remove everything else, retain only this domain, and this domain would hold and would perform at least some function, like bind ATP, whatever. So this is like an experimental approach to the definition of protein domains. Another approach is that domain is something that folds more or less independently of the rest of the protein. So it's like some compact part of the protein when we look at its 3D structure. And then, of course, a lot depends on how do you define what is compact, what is independent, because the proteins are not like beads on a thread. It's not like you have one domain, the other protein, and like long linker in between. It's too good to be true. It's actually highly intertwined. And then, when you define a structural domain, actually a lot depends on like parameters that you put into this definition. What is compact and what is separate from the rest. Like in all cluster analysis in whatever other area. And the third definition, which I actually like the most, is that protein domain is something that evolves independently of the rest of the protein. And this is actually the definition which is used in databases like EPAM, protein families. It's actually the database not of entire proteins, but rather of protein domains. And protein domain is here is defined as subsequence of a protein which you observe in many different contexts. Which you observe in the company of other fragments that also are independent domains. So domain is something that is it's a unit. It's never destroyed by evolution, but it's the unit that you can find in different contexts that evolves independently of the rest of the project. And now again, here a lot depends on your exact definition, because what I said is an idea. It's not an exact definition. To make it exact, you need to write a program, and you'll have lots of parameters in this program. And so with one set of parameters, you'll have one definition of one set of domains. With another, you'll have another set of domains. But basically, that's the idea. The evolutionary domain is something that evolves independently. And actually, if you go back in the history of bioinformatics, it took a while to realize that this partial similarity of domains actually might create a problem for annotation. And it's one of the nicest examples. It's very old. This is the output of some absolutely obsolete program. I don't even recall its name. It's not important. What is important is that this protein, MG262, it's actually a gene, the protein encoded by a gene in Mycoplasma genitalium that has been the first completely sequenced bacterial genome. And there was a protein, this one, which has been annotated as a DNA polymerase homolog. Because it was homologous, it was similar to several DNA polymerases from 3 different bacteria. Actually, E. coli, Camus aquaticus, and Bacillus, whatever, I don't remember. I don't know. Now, the funny thing is that this domain actually has no polymerase activity at all. Because the clone of fragment, which is the DNA polymerase fragment which has been used in genetic engineering and molecular biology for years, this is a domain which has a polymerase activity. And the N-terminal domain of this protein is actually an azonuclease whose function is spell checking. It removes incorrectly inserted base. So it recognizes mispaired base pairs and removes the incorrectly inserted nucleotides. So this entire annotation based on based on local similarity is a nice example of of people misled by domain similarity, assuming general similarity of functions. Okay, now actually in new incarnations of BLAST, you probably would not run into something like that because when BLAST starts running, the first thing it does is to compare your query sequence with domain databases. So if your protein has some non-trivial domain structure, if it consists of more than one domain, then you'll get output something like that. So you see that it's ribose repressor probably or some transcription factor, and what your program tells you is that it has a DNA binding domain here, and actually it has a domain which is annotated as a periplasmic component of transporter system. Now, it's a nice story because what has happened is that indeed the substrate binding domain of this family of transcription factors, indeed its origin, I mean, it originated from a transporter, from a periplasmic substrate binding domain. When this substrate binding domain merged somehow with helix to helix, with the DNA binding domain, what happened that family of transcription factors was born. Things like that happen a lot. I mean, one of my former students has a collection of families like that, families of transcription factors that arose by a merger of a DNA binding domain with either a transporter protein or maybe an enzyme. And because what you need from transporter protein or an enzyme in this case is just the ability to bind a specific substrate and probably change its conformation. And then this change of conformation influences the ability of the DNA binding domain to actually recognize its binding sites and bind to that. So a naive person actually might, well, you have lots of annotations like periplasmic binding protein, like here and here. So again, this is an example when domain similarity might mislead you if you're careless, even in a good situation when the database actually provides you with a complete scheme of the domain organization of protein you're looking at. But again, there is a database of domains, and now there's another important issue, which is often underestimated by experimental biologists. True experimental biologists just never touch computer unless, I don't know, read email or I don't know, read Facebook or whatever. Spoiled experimental biologists have heard that computer is useful for something. And then they run, for instance, they run BLAST. They see protein which is similar to the one they are studying. And then they assume that, okay, well, let's assume that the similarity is full length. So there is no issue of partial similarity of domains, it's full length similarity. Okay, when two proteins are similar over the entire length, well, probably they're doing, well, they must be doing the same thing. And again, that's not necessarily so. because there are two reasons for homology. Well, there are two ways, actually more than two, but let's start with the two basic ones. There are two ways in which protein homology, two ways in which homologous proteins may arise. Orthology, which is divergence following speciation. You had some ancestral species, you had a protein in this ancestral species, the species diverged into two, the proteins start to accumulate mutations, or there are two different proteins now, but they have common origin, they arose by speciation. In most cases, they do retain function, but not necessarily. And the other possibility is gene duplication within one genome. And then you have two copies of the gene which encode identical proteins. This situation clearly is not stable, so it might easily be that one of the proteins, or one of the genes will immediately get lost. But sometimes both copies survive, but you don't need two copies of the same thing. So they start to diverge in function. And in case when two proteins are related through duplication, indicates that there should be something different in their function. Which actually, well, that's, as said, it's fairly nice and simple and good and useful. The problem is that this speciation and duplication events might occur. Okay. Well, let it be here. So, oh, cool. So you have two species. You have an ancestral species here, ancestor. And you have two descendants. And descendant two. Okay, so what might happen? You have a gene here, you have a speciation event here, do you see it, or should I try to get rid of the letters? Or is it okay like that? Okay, thanks. You have a speciation event here, or you have two different genes. They have diverged during the speciation event, so they are orthologous. They probably retain the function. Now, retaining the function is not part of the definition. I'll give you examples later. But it's rather an empirical rule that orthologs normally tend to have the same function. Now, what might happen is, this protein, this gene, has duplicated here. So this is a duplication event. So now we have genes 1 and 2, and now we have genes 1, 2, and 3. Now, 2 and 3 are clearly parallel, because this is duplication. But which of these, 2 or 3, is the ortholog of 1? Why 2? come on, it's duplication, it's symmetrical, you don't know what is the original one. You just have two copies. I mean, it's segmental duplication. You had a gene, and now you had two genes in the same place. Which of them is the original one? Nobody knows. So the trick is that actually it should be symmetrical. And then both 2 and 3 are orthologs of 1. They're called co-orthologs. Now, in this case, we do know. Okay, well, let's look the other way. Let's look at it the other way around. So suppose, again, you have your two species, and you have a gene here and a gene here. And these genes are homologs, so they had common ancestor somehow, somewhere. Can you guarantee that these two genes are orthologs? They're homologs in different species. Does it necessarily imply that they are orthologs?
No, because of convergent evolution.
No, well, we don't believe in convergent evolution. And okay, so suppose I told you actually that they are of common origin. So, I mean, yeah, okay. Let's assume that they really are homolog. So their similarity is not due to convergent evolution or skewed amino acid composition or whatever. So if I tell you that one gene, I have a pair of genes, one gene in one species, the other in the second species, I tell you that these genes are homologs. Would that necessarily imply that they are orthologs?
There might be a scenario when there was a duplication event because before divergence event, then all these duplicated copies became divergent.
Yes. Actually, what would happen is that I had a duplication event here. So we already have two genes by the time of speciation. But then, either you just haven't sequenced these genes in this species yet, or they might have been lost during evolution. So what might happen is that, well, by definition, during speciation, all genes are inherited by descendant species. But then, during evolution of this species, so by definition, we have to have this fork here and this fork here. During speciation, we assume that during speciation, nothing is gained and lost. We just have two genomes copied exactly as is to descendant species. But these copies might have been lost during evolution. And so what do we see in this case? We see that we have homologous genes. They are similar to each other. They are common. The level of similarity is sufficient to assume that they really are homologs. But they are not orthologs because the duplication preceded the speciation. Think about like alpha globin of human and beta globin of, I don't know, mouse. They're clearly homologs, but they never, they are not orthologs. But in this case, actually, human has both alpha and beta, and mouse also has both alpha and beta. So the duplication into alpha family and beta family has occurred in some, I don't know, common ancestor of vertebrates, probably. But again, as I said, these copies might have been lost. Okay, now this is very nice when you do know what happened. When you do know the species tree and basically, well, I draw the trees, the phylogenetic trees of individual genes. The problem is that normally you don't have this scenario, and you have to reconstruct this scenario from your phylogenetic trees. So normally, you have something like that. Well, let's assume that you do know the species tree. So the species tree, you have three species. And they are related like that. So you have species A, species B, and species C. Now let's assume that you have a phylogenetic tree like that. Well, let's start with a simple story. The simple story is here. So this is your B, C, A. So this is gene number 1, gene number 2, gene number 3, gene number 4, gene number 5, and gene number 6. And they come from, so A and 5 come from species B, 2 and 6 come from species C, and 3 and 4 come from species A. So this is your gene tree, something that has been produced by your whatever phylogeny reconstruction software. And this is your species tree that you just know from somewhere. So let's assume that the species tree is given. Now, can anybody draw, fit this gene tree into that species tree? What has happened? How did this gene evolve within these genomes? Any takers? Okay, well, let's have a break. And we'll reconvene in 5 minutes in 7.5. And then I'll ask for a volunteer actually to do this mapping.